Hi everyone, this is my first attempt at a recorded PowerPoint presentation to make it more like we were actually sitting in class with a lecture. This is the PowerPoint on microbiology, which we had started briefly when last we met, right before spring break. So I'm going to pick up at the beginning, even though we had done a few slides, but I'll start at the beginning. With the microbes, the term microbes or microbiology really refers to any living thing or organisms that you can't see with your naked eye. And we had talked about the different categories, but we're going to focus in class on bacteria first, and then we'll talk about viruses. Many infectious diseases, anything that's infectious means you can catch it from someone else, as we're more than aware of right now, are caused by one or more types of microbes. And so we're going to be talking about them. As I go through the presentation, when we get to bacteria and viruses, I'll be talking about a lot of different infectious diseases. You do not have to know specific about the diseases, but more focus on the characteristics of bacteria and then viruses as we go along. So this is just a category of what different microorganisms are. And again, we're just going to focus on bacteria and viruses. We're not going to really talk in detail about fungi or protists. So I'm going to start first with bacteria. And bacteria are prokaryotic cells. If you remember back at the very beginning of class, we talked about the difference between prokaryotic and eukaryotic. We are eukaryotic. So bacteria are much, much smaller than our cells, only about 1,000th the size. All bacteria are single-celled organisms, so they're unicellular. Although sometimes they clump together and form colonies, they still exist as individual cells. They're the smallest cells that we are aware of. Their structure is much simpler than eukaryotic cells. They have no internal membranes. What that means is they don't have organelles like a nucleus or mitochondria or chloroplast. They do have DNA, just like we do, but it's not organized inside a nucleus. It's just clustered together inside a cell. This slide just gives you an idea of relative size. Up here we see under a microscope, magnified at 200, a human hair. And so this little cluster here is a comparison. And these are eukaryotic cells, red blood cells, white blood cells, yeast cells. And here, this little cluster here are bacterial cells, E. coli, and then little tiny ones inside of there are viruses. So you can see how much smaller bacterial cells are compared to human cells. So this would be an E. coli versus a red blood cell. And then viruses are even markedly smaller. This is another good picture because remember when we did the cheek cells, the very first lab where you scraped the inside of your cheek and you looked under the cells? When we did it, ours were stained blue, but these are cheek cells that are stained pink. And so this is one whole cell, human cell here. And then these little dots inside are streptococci. So like if you had a strep infection, and you can see how tiny they are compared to the human cell. This is also another, this is a scanning electron microscope picture. And this is a tip of a surgical needle magnified thousands of times. And each of these little yellow things are an E. coli. And so you can see how many there are just on the tip of a needle. This is not to indicate that surgical needles are usually contaminated with E. coli, but this is just using it to give you a relative reference of size. Bacteria are characterized by a number of different criteria. And one of the criteria is shape. And sometimes that's incorporated in their name, not always, but sometimes. So a spirillum is like a spirally shaped or twisted, kind of looks like a piece of pasta or something. Bacteria that are bacilli are kind of rod shaped. You've seen pictures of that. That picture I just showed you of the E. coli. E. coli is a rod-shaped bacteria. And then cocci are circles. And they do sometimes form chains, but each one of these little circles is one bacterial cell. So things like Streptococcus, Staphylococcus, they're all cocci. So they're circular bacteria. The structure of a bacteria, all bacteria have a cell membrane, pretty much exactly like our cell membrane. That is, it's a phospholipid bilayer with proteins stuck inside of it. But bacteria also have a cell wall. And we talked about the cell wall of plants being made of cellulose, which is a polymer of glucose. Bacteria cell walls are made of different substances than plant cell walls. The primary component of bacterial cell walls is something called peptidoglycan. And peptidoglycan is a type of carbohydrate. This composition of cell walls is one of the criteria that is used for determining how to treat different bacterial infections. And depending on the composition, bacteria are divided into what's called gram-positive and gram-negative. I'm going to talk about them in more detail, and then we'll go into other external structures of bacteria, flagella, which is like a tail, and pili, or little fuzzies. And I'll show you pictures as we go through. So bacteria can be classified as being either gram-positive or gram-negative. This is based on a lab test that's done, and it was developed by a man whose name was Graham, and so it's named after him. And it's still used today for doing classification of bacteria. This picture here shows both gram-positive and gram-negative. Gram-positive bacteria turn like a purple color, and gram-negative are pink. I'm just getting used to doing this. So if you look at this picture here, the pink ones would be gram-negative bacteria. The purple ones would be gram-positive bacteria. And the difference between a gram-positive and a gram-negative is the composition of their cell wall. So a gram-positive bacteria has a one-layer cell wall that is made up just of peptidoglycan. And so that's what's facing the outside world. And when you do a gram stain on a gram-positive bacteria, they turn that purpley color that we saw. And this is just showing what that would look like. So here's our bacteria. And if you cut out a little piece of its outer surface, this would be the membrane, phospholipid bilayer. And then on top of that is the cell wall. And this one is all peptidoglycan. And so the stain touches the peptidoglycan and turns it purple. And that's why they are gram-positive. The gram-negative bacteria have a more complicated cell wall that has two layers. They're the ones that turn pink. And if you look at their cell wall, it has, this is the membrane again, and then this is the peptidoglycan part of the cell wall, but then exterior to that, that is facing the outside, I keep using my hands and realize you can't see my hands, is another layer that's more like a membrane. So when you put the gram stain on those, it doesn't get to the peptidoglycan because this other layer is protecting it. And so it doesn't stain purple because of that outer layer. So that's a gram negative. And the reason this is important at times, let me just go back to that slide, is that with a gram positive bacteria, some antibiotics work, like penicillin, for example, works by disrupting an enzyme that makes the cell wall, the peptidoglycan cell wall. So if you have an infection with a gram-positive bacteria, any antibiotic, such as penicillin, that inhibits the ability of the bacteria to make its cell wall would be effective on that infection. If it's a gram-negative bacteria, that outer layer kind of coats the peptidoglycan, and so it doesn't get to it, and therefore it won't be significantly killed by some antibiotics that affect cell walls. So knowing whether your infection is due to a gram-positive or gram-negative bacteria would influence the antibiotic that might be used to treat that infection. Another external structure on a bacteria is many bacteria have flagella, which are these tail-like appendages, which enable them to change direction and move. We talked about flagella a little bit. The only human cell that has a flagella is a sperm cell. And then some bacteria have this kind of like fuzzy stuff coating them. And that's called pili, P-I-L-I. And that's like a fuzzy coat on the outside of the bacteria. And we'll talk about that a little bit more in a minute. Okay, now we're going to talk a little bit about the inside of a bacterial cell. They have cytoplasm, like we do, like the fluidy part. They have DNA, but instead of having a lot of chromosomes, they just have one. So their DNA, it's called circular. It's kind of like this thing like that, all attached together. They do have some structures, but not big membrane-bound ones. They have ribosomes, because remember, ribosomes is where you make proteins. And the bacteria do have to make proteins, just like we do. They make enzymes. Some bacteria have the ability to go into a dormant stage where they form something called endospores, which are spores inside. I'll give you a picture. A picture of some bacteria with endospores. It's kind of like this little dark area in here. And that enables bacteria to be in a kind of dormant phase. One that you may have heard of, an example of this is, if you remember a number of years ago, there was a period where there was anthrax being mailed in envelopes to different people, such as, these are two of the letters, a senator, this is a newsman. And what was in there were these endospores of anthrax. And anthrax is caused by a bacteria. And normally that bacteria is found in the ground. And if you come in contact with it, you get what's called hand and mouth or foot and mouth disease if you're a cow. And you get like a skin infection. So it's not real serious, but it can form these endospores that are these dormant, and it almost becomes like powder. And that was what was being used in these letters. And in those cases, it's not really just touching your skin, but you're inhaling these spores, and they get into your lung or even beyond that and give you a respiratory infection. And that can be much more serious. And so there were several people that died from this infections from inhaling the spores in these envelopes. And so the spore formation is something that when you hear of like biological warfare where they disseminate spray of bacteria and stuff, that's one of the things that could be used for that. But not all bacteria have that ability. Clostridium botulinum, which is a obligate anaerobe. So it can only grow in poor oxygen environments. And it produces a toxin, which is a neurotoxin, which means it affects the nervous system and the ability of the brain to cause muscle contractions. And so it's a really potent toxin. It's a very toxic bacteria, but luckily it only grows where there's no oxygen, so it's not present in large amounts around us. There have been a couple incidences of people dying of botulism by consuming canned food that had been improperly canned so that there were still bacteria growing in it, and that is a poor oxygen environment inside a sealed can. And so there have been some cases of botulism from that. Luckily, the toxin is destroyed by heat. So if it's something even present in the canned food, as long as that's heated before you would eat it, it wouldn't be a problem. That bacteria, Clostridium botulinum, is the source of the active ingredient in Botox. So Botox actually stands for botulinum toxin. And because it is a neurotoxin that causes the nerve not to cause a muscle to contract, it's used probably most commonly cosmetic kind of treatments to reduce wrinkles, which are caused by a muscle kind of contraction. It's injected in very small quantities at local spots. It's also been used sometimes for cerebral palsy, where you get overconstriction of muscles, which makes walking and some other movements difficult, so that they can inject small amounts of Botox and relax those muscles and make for easier movement. So, but that's actually extracted from these bacteria. They're not actually injecting the bacteria, they're injecting small amounts of the purified toxin from those bacteria when you get a Botox injection. Another criteria for classifying bacteria is, we talked about their shape, we talked about whether or not they require oxygen, and the third one we're going to talk about is nutritional requirements, whether they need to eat, so to speak. So some bacteria are autotrophs, and autotrophs are like plants in some ways. They can make their own food. So plants, for example, use energy from the sun for photosynthesis and make glucose. And some bacteria have photosynthetic capabilities or can use other energy sources to make their own food, so that they do not require essentially any outside organic compounds. Other bacteria are heterotrophs. We're heterotrophs. We need to eat organic compounds. So we need to eat carbohydrates and proteins and fats. And some bacteria are also heterotrophs. So ones that grow on decaying food matter, which are called decomposers, they are eating whatever they're growing on. So if you have bacteria growing on your food that's going bad, they're actually eating the organic matter in your food. All of the pathogens, that is all bacteria that cause disease, are heterotrophs, because the reason they're infecting you is because they're looking for their nutrition. And so they're eating compounds that you're producing. So they also need either carbohydrates or proteins or fat to survive, just like we do. The final category we're going to talk about is how bacteria reproduce. So bacteria are asexual. That means there's not girl and boy bacteria. They just make a copy of all their genes and then split in two. That's called fission. So that's the way that bacteria reproduce. This is a picture of a bacterial cell dividing. So once it splits, they'll have two bacteria. Bacteria that way, as long as there's sufficient resources, space, food, etc., they can grow pretty rapidly because they just keep dividing in half and doubling their number in a short period of time. One of the problems with asexual reproduction is that you're not changing over time. There's always, your genes are staying the same. So that can sometimes make you more vulnerable. If there's some kind of a harmful mutation, it gets passed on to everybody. Not everybody, every bacteria. And so bacteria also have a way sometime of sharing genetic information. I'm going to have to skip that animation because it's not going to work. And that's called through conjugation. And so conjugation is not really reproduction, but it's sharing genetic or genes. So suppose one bacteria have these genes that give them some good ability. They can share them with another bacteria of the same species by doing what's called conjugation. And here's the bacteria. These are all those little pili that we talked about. See how fuzzy that is? And this bacteria forms what's called a conjugation bridge. This is actually a little tunnel that connects it to another bacteria. And it can actually, through that tunnel, pass through genes. So that if it has a gene that it gives to this bacteria, then that bacteria now has the ability to do whatever that gene is. And so that's one of the ways that bacteria can share genetic information even though they don't reproduce sexually. Strep throat, scarlet fever, pneumonia is one. Pneumonia is an inflammation in the lungs. It can be either bacterial or viral. So that's non-specific. We talked about anthrax. Legionnaire's disease is one that first appeared a number of years ago, and it was at an American Legion convention in Philadelphia, so that's why it's called Legionnaire's disease. You might have heard of this. There's been a few other outbreaks. It seems to be a bacteria that can live in air conditioning vents and circulation vents. And so the Legionnaires were also at a convention in Philadelphia and staying at a hotel and going to meetings at the convention. So they were kind of all inhaling that air. There have been a couple other outbreaks on cruise ships. Skin diseases, any kind of like pussy infection is usually bacteria. We'll talk more about that in a minute. You can get some GI infections, like some of the food poisoning, like the E. coli was in the news a number of times, salmonella. Those are all bacteria. We talked about botulism. Meningitis is another one that meningitis technically means inflammation of the meninges, which is the membrane covering your brain. And so it can either be viral or bacterial, depending on what the source of the inflammation is. Diphtheria, typhoid fever are also bacterial. Lyme disease, you may have heard of. That's named after the town it was first noted in, Lyme, Connecticut. That's one that is tick-borne. And so the tick doesn't have the disease, but it's called a vector, which means if the tick feeds on an animal or person that has the infection and then goes to another one, it can transfer it. And so it can be transferred like between mice, deer, and humans. It causes a kind of characteristic rash called a bullseye rash that is usually indicative of Lyme disease. It was pretty widespread in the area that I lived in New Jersey. Ticks aren't as common here in southern Arizona and central Arizona, so you don't hear much about Lyme disease, although there are a few other tick-borne diseases that are around. Bacterial diseases in general are the only ones that can be treated with antibiotics. And again, depending on whether it's gram-positive, gram-negative, that would influence the specific antibiotic given to treat a specific infection. But all antibiotics are geared towards bacteria. They do not work on viruses. So antibiotics work, a number of them work by inhibiting cell wall synthesis. And that's what I mentioned before, penicillin and ampicillin do that. And so those are most effective on gram-positive, whereas bacteria that have other effects, like on protein synthesis, on making proteins, would work on gram-negative as well. You probably have heard that bacterial resistance to antibiotics is a big problem now. And what that means is that bacteria have mutations. Some bacteria have mutations that make them resistant to a particular infection. So say, for example, antibiotic works by inhibiting an enzyme needed for cell wall biosynthesis. And so when treated with that antibiotic, the bacteria can't make their cell wall, and therefore they can't survive. But suppose some bacteria has a mutation that their enzyme is made slightly differently, and that antibiotic doesn't affect their ability to make the cell wall, then they would not be hurt by that antibiotic and would go on and be able to reproduce and make more bacteria that have that same mutation. And that's what leads to bacterial resistance. So here's an example here. So suppose you have a normal bacteria that's not resistant. So you get an infection, the bacteria start multiplying, and then you take the antibiotic and it kills them, and you go on and you're fine. But suppose one of those bacteria has a mutation that antibiotic doesn't kill it. So when you take that same antibiotic, it's not affected, and it goes on to reproduce. And over time, more and more of the bacteria are antibiotic resistant, because the ones that aren't are being killed by the antibiotic. And so antibiotic resistance is a big issue. The one that you might have heard of is MRSA, which is MRSA. And MRSA stands for methicillin, which is an antibiotic, resistant Staphylococcus aureus. Staph infections are very common. Anytime you have a cut that it gets a little pussy like this, that's magnified, so it looks worse. But that's usually a staph infection. And staph bacteria are kind of everywhere, so it's not uncommon to get a staph infection. And usually they'll go away, or if you take an antibiotic, it will get rid of the staph infection, and it's not that serious. However, MRSA are staph that are resistant to most antibiotics. And that means that when treated with antibiotics, it does not affect their growth. And so you can get a much more serious infection. It can become systemic, which means it can spread throughout your body, cause liver damage, kidney failure, and can possibly be fatal. This is a petri dish. This is what you're going to be doing in lab this week. And you can see these are little pieces of filter paper soaked in antibiotic. The yellow stuff is bacteria growing. And you can see that the bacteria can't grow close to the antibiotic because it kills them. But here you have some that are much closer that aren't being as destroyed by the antibiotic. So MRSA is an increasingly serious problem. It can be spread easily. if you have a cut or a scrape on your hand and you have a staph infection and you pick up a towel and somebody else can get it. So these are some of the guidelines, which are very similar to the guidelines we're talking about now with wash your hands frequently. And if you have any kind of cut or scrape on your hand or any area that you will be coming in contact, you should cover it up to prevent the spread of MRSA.